Hi, class. So I hope you're doing well in these extraordinary times. Hope you're staying healthy and doing well with the practice of social isolation or social distancing. I guess they're about the same thing. So for this week, we're going to talk about Bioart. I hope you enjoyed the readings and the viewing. I think the CAC TED Talk is pretty good as a supplement to his text. So we're going to talk a bit about. I'm going to show you a bit of his work during this lecture, just again just to supplement your reading, and then I'll be interested to hear your responses. And remember, your response. If you need another day, maybe even 2 days, it's fine to take a little bit more time if you want to work on your reading response and have it in on Friday. That's totally fine with me. That way, maybe you can intersperse some of your ideas that come from the other works that I show you today while we go through Bioart. So I hope you find it interesting. Oh, the other sort of little announcement, housekeeping thing is that it seems we have another break, what they're calling reconfiguration. So next week, this hasn't been finalized yet because it's kind of unclear. I haven't heard from the department. Once I hear, I'll definitely give you an update on the website. But it looks like we're probably going to be moving things back, which is fine. It shouldn't affect our class too much, but we'll finalize that as we go forward. So without further ado, Bioart. So in its basic definition, I mean, like the basic understanding, the term is coined by Eduardo Keck. Bioart or transgenic art. It's a little loose, but basically it's artists that are working with what you might want to call the biological building blocks of organic life. So these include, we're going to talk about animals. This is an animals. We're going to focus mainly on animals, but we're also talking about plants, and we're also talking about any form of organic life. That means plants and animals, humans included. So these basic building blocks are being used basically as the medium itself. So rather than using paint, sculpture, video, or the body itself as a performance, like in body art, Bioart or transgenic art uses that works on the somewhat smaller scale. So like genes, proteins, tissue cultures, and that sort of thing. So that's the basic. That's the basic definition of bioart, and I think it's quite clear how it implicates the animal, which is our focus in this class until we get to the larger scale issues towards the end of the semester. And so bioart taps into it's a very recent thing. It needs breakthroughs in biogenetic technologies that only come in the 20th century, later 20th centuries. Excuse my coffee sipping. I'm doing this in the morning. It does tap into a kind of a fundamental artistic impulse, a mythic one, which is to make a work that's so good that it basically becomes alive. It becomes life, where art and life completely collapse. And one of the primal scenes for this fantasy, for this myth, which maybe is no longer a myth after these scientific breakthroughs of the latter part of the 20th century, one of the primal scenes for this desire to make a work that becomes life itself is Pygmalion, the Greek myth. And there have been all sorts of variations of this. I think like Edgar Allan Poe has a reverse example of this. I'll tell you about it in a second. It's a great little short story if you want to read it. But the original Pygmalion myth is basically about a sculptor who falls in love with his creation, falls in love with his sculpture, and kisses it, and it comes alive. So basically like the artist's kiss, breathes breath into this, and the statue becomes flesh and blood. So the work as a living organic thing. The Edgar Allan Poe version of this is actually in reverse, where he... what is the name of this short story? We'll have to look it up later. But it's one of Poe's short stories, and he paints the portrait of his wife. And the more he paints the portrait, the more beautiful the portrait is. The more the wife starts to wither away, and towards the end, she dies when the painting is complete. So it's kind of a reverse, in some ways, a reverse of the Pygmalion story. But that's the basic impulse. That in some ways, you could find it in a number of corners within art history. This idea, this desire to make a work that's so good that it'll become a living thing. When we talk about this restricted to animals, to non-human animals, there's a history of this too. So this is a print, an etching. I think it's an etching. It blew me away. Last year there was a Delacroix retrospective at the Met. It was such a gorgeous show, and this print in particular was just mind-bogglingly beautiful. It's his royal tiger. Delacroix is he's a romantic painter during Romanticism, and he became, one of his staples was what's called animalier painting. So the representation of animals. So he has beautiful, wonderful animals in his paintings and in his drawings and his etchings. And so this is an example, just the one that blew me away. He never actually saw these animals in the wild. So this tiger he would have seen in the zoo in Paris around 1829. But you should note how it looks like you're getting the tiger in its natural habitat, kind of, with the hill in the background. He looks like he's in the wild, right? So that's a staple of animalier representation, showing animals not in confinement, but in the wild as this feral, beautiful, imposing creature. And so this is an example of animalier painting or drawing. And the notion with these animalier works is that while the artist is able to represent the animal, at least in the West, there's this long-running idea that the creator with a capital C is God, and that these animals are made by God, right? And so it's foolhardy, maybe even hubristic, maybe shameful for an artist to try to replicate the thing that God made within the history of Western art history. And so there's always this uneasiness of the artist making a work that's an animal that can only then, in the eyes of him, or sometimes her, and their contemporaries, can only be pale copies of the actual divine creation. Right? So it's always understood there's a long-running history of this, and some artists encode this in the work to make sure they're showing that they don't think they're really representing the animal. They don't think they're really sort of creating something here. They're just a pale imitation of the true creation. Right? So this is a long-running history. And the reason I bring this up with bioart is because this is one of the interesting questions. What happens when, and this will happen in the course of the 20th century, what happens when we no longer limit ourselves to simply representing life, with the buffering, with the caveat that this is a pale copy of a divine creature, that the maker is not the artist, but the maker is God? What happens when we move away from that and we can actually start generating forms of life ourselves? We can actually start playing with life. We can actually start creating new forms of life. This is one of the big questions with bioart. And so I do want to stress here, and when you read anything about bioart, it's often noted that actually this is not totally new. And in fact, the practice of breeding, which is quite old, the practice of selectively breeding like cattle or chickens. I read this bizarre case of a scientist. This is in the early 20th century. Of course, breeding goes way back, right? But in the earlier 20th century, there's a scientist in Israel who breeded a chicken to not have feathers, so that basically the chicken would be easier to process, right? So this Federalist Chicken, which you can Google, I didn't want to show you an image on the PowerPoint because it's just kind of, I don't know, they look naked and obscene and it's kind of disturbing. The reason why they're bred this way is so that they're basically easier to process, right, for food. So we'll talk a little bit about how these works of art implicate food towards the end of this lecture, but I do want to just stress the point that actually the practice of breeding, and if not creating new life, sort of coercing new forms of life, life that wouldn't have happened otherwise through natural evolutionary processes, unless you assume that human breeding is part of evolution, which actually you probably should, that this is not totally new. But with that caveat, these techniques, these biogenetic techniques, these transgenic techniques, ones where you can actually go in and start tinkering with the genetic makeup of life, that is going further than breeding. That's definitely going further than breeding, and it's probably a difference. It probably is a difference in degree to breeding, and it does allow for some pretty groundbreaking developments in the history of science, in culture, and then of course in art itself. So I'm just going to show you two groundbreaking examples. The first less groundbreaking, less well known than the second, but still quite important. So this is in 1988. This is the Oncomouse. This is the first animal to ever be patented. And it was a big controversy in the 80s because I think you can understand a lot of people were really uneasy with patenting an animal. And it was patented by Harvard University in 1988. So it's a special little mouse. It's also a little disturbing. The mouse is transgenically predisposed to have an activated oncogene. This is why it's called the Oncomouse, and that's how it was patented and trademarked, Oncomouse, which is why it's in uppercase there. And so this activated oncogene significantly increases the likelihood that the mouse will get cancer over the course of its life. Why would you want to do this? Why would scientists want to do this? And before this, actually, scientists bred mice in all sorts of ways in order to try to get certain traits that they could then study. Well, the reason is that if you're doing research on cancer, it's much better to have cancer growing within a live organism. So a mouse is not a human, and of course there are all sorts of problems with mice being tested on for human research. But it can safely be said that it's better than studying the cell line in a petri dish under a microscope, right? Because you're actually seeing what the cancer is doing within a living organism, within a mammal, which is significant since humans are mammals. And then the idea is that you can then parlay that into cures or treatments or therapies for human diseases. So in this case, it would be cancer. So here we get back to our animal rights lecture, where this is very tricky stuff, right? Because there's a part of me that thinks, it's just horrible to create a living thing, a mouse, that's going to be predisposed to get cancer and probably have a painful death or suffer in some ways, right? And that it's difficult to think about it. But on the other hand, let's say this one mouse leads to a treatment for cancer, right? Benefiting millions of human beings who are sick. As a strict utilitarian, you would likely say that this is justified, right? That it is actually an ethical good, that the good outweighs the harm. So these are challenging, difficult ideas that we're dealing with. But for our purposes today, for Bioart, this is just a nice example of transgenic work, where you actually go in. Here you go into the embryo. I think it's into the embryo to to distribute or to activate this oncogene, or to insert it into the genetic code of the mouse, so that once it's born, it will have that trait. It will have that activated oncogene. So that's the Oncomouse. More famously, this is in the 90s, and there are some people that have written. I was just reading an essay yesterday. Someone saying there are three major developments within the history of humanity in the 20th century. The first is the atomic bomb. So the idea that we can destroy ourselves. We're the first species that can completely destroy the planet. That has some serious ramifications for evolution, right? It would just end most evolutionary processes, maybe all. The other is cracking the human genome, the Human Genome Project, so like understanding the basic building blocks of life, of all life, plants, animals, human and non-human, that were that DNA itself is made up of these four components. Then there is Dolly, and the first mammal to be cloned, and this happened in 1997, and this was a huge sensation. I'm not sure if you've heard of Dolly, but it was a major story. So this was the first mammal to be cloned. So there was a cell from the breast of a ewe, a female sheep, that was implanted, that was fertilized with another sheep, and then there was a surrogate mother, and the embryo that it went in was stripped of its DNA. I think this was the crucial step, so that the resulting birth would be like a live birth. It would just be a normal birth of the sheep. The sheep would be an exact clone of the mammary gland, mammary cell that it was taken from. So what you had, in effect, Dolly was the first animal that was an exact replica, DNA replica of its source, right? So this was the first mammal to be cloned, and there had been smaller animals that had been cloned before. I think years back there was a frog that was cloned, but there was something about the mammal, and there was something about Dolly, and this is a bit gross. The eco-feminist in me finds this kind of gross, but the doctor, one of the scientists who worked on this, named the resulting sheep Dolly after Dolly Parton. So this is in reference to the fact that she has large breasts. So I find that kind of gross, with the idea that the cell comes from the mammary, the breast from a ewe. So that's where the name Dolly comes from. But this was a big deal because this was the first clone of a mammal, and if you read. There was a really wonderful, it's only 12 minutes long, so it's not too long. I recommend it, and I'll probably embed it to the website once I upload this lecture. It's a 12-minute New York Times thing about Dolly and sort of the news that was generated at the time. It might be hard to believe because I think who just cloned their dog? Barbara Streisand or something? She just cloned her dog who was dying, so she wanted another, like another dog exactly the same. Back then, cloning was quite scandalous. It created quite a big controversy. And this is, again, if we talk about the history of Animalier paintings and religion and God as the creator, this was part of that discourse, right? There were a lot of people they were against this because they thought we're tinkering with life itself, and we're acting like God, and that actually this is wrong, right? This is wrong at like a theological level. There were others that were maybe a little bit more science fiction dystopian, where they thought, okay, well, we can clone sheep. Well, what's next? We're gonna clone humans, right? What if you have someone? This is in that New York Times video, which I encourage you to watch. There was someone at the time who said, yeah, what if Hitler would have been able to clone himself, and then you'd have an army of Hitlers or something like that, right? So it's pretty wild how new scientific breakthroughs, and there are sort of legitimate concerns, right? Like one of the most pressing ones for me is what happens. What if you clone something into existence and there's something that goes wrong, right? What if the very act of cloning leads to a life of suffering or some sort of impoverished life? And it should also be noted that Dolly was not the first that they tried, so there were lots of failures to clone. So you have basically, surrogate mothers who are maybe dying, or surrogate mothers who are giving birth to sheep that die right away. This is like Dr. Moreau territory, where we're sort of, there's a fine line between, one could argue there's a fine line between proper scientific work and then like the crazy Frankenstein kind of work, right? Where you're creating these monstrosities. So there are legitimate concerns, but then of course when you have scientific breakthroughs, you also have more, let's say, far-fetched concerns, or people just overreacting. And this also got into the controversies over stem cells, which started around the same time. But for that, I'll advise you to watch the video. So Oncomouse and Dolly, two transgenic creatures in the late 80s and in the 90s, that are seminal moments in this history. And this will then lead to artists like Eduardo Cap, who you read about, working with transgenic techniques in biotechnology within their works of art. So I'm going to go through these somewhat more quickly, because otherwise this lecture is just going to end up being way too long, and you're gonna be talking about these works in your writing. So I won't dwell on them too long. But his first transgenic work was Genesis. You'll remember from the readings. And so here, this was transgenic bacteria. So going in and splicing the genes of a bioluminescent jellyfish into these bacteria, so that the bacteria would themselves, under blue light, become bioluminescent, so basically glow in the dark, right? He also encoded a passage from the Bible, from Genesis, which is where this title of the work comes from, Genesis, and the famous Dominion passage, right? So he translates that into Morse code, and the Morse code gets translated into the GACT, the 4 amino acids of DNA. Then he sends that off to a lab to make the DNA itself, and it gets inserted into the bacteria. So that's why you're seeing all these letters here, the GTAC. That's the basic building blocks within our DNA, the double helix structure, and then let man have dominion over the fowl of the earth, and blah blah blah. This is from Genesis. And then there was another interactive component. Kak is into these sort of transgenic works, but he also thinks that they should be part of this larger discourse, or this feedback, or this larger discussion within the public. I think that's really clear. And so in this one, you could also go in remotely from online and start tinkering and with the blue light, and the light itself would then create mutations in the bacteria. And after the course of the exhibition, that means that the very genetic material of this bacteria were altered, which means that the text itself, the passage, was also altered. And so he had that translated and uploaded to the website. And the idea is that through this interaction, through the processes of genetic mutation, this famous passage from the Old Testament, from Genesis, gets altered. So that's the basic premise of this work. I hope you'll talk about it a bit in your responses. But then his most celebrated and notorious work is the Gfp Bunny from 2000. So here he is, not with the bunny, not with Alba. The bunny was named Alba. Because you'll have read that once he actually does the work with a laboratory in France, with the artist in collaboration with, or helped by, the artist Louis Beck, who in his own right is important for the history of bioart, though I don't talk about him too much in this lecture. We may end up talking about him later on in another lecture. Excuse me. This is Alba. So Alba, this is not what, this is, no one is really sure because no one has seen Alba. But Alba would have been a white rabbit who would have been a transgenic rabbit, so like the bacteria in Genesis, although this is now a mammal, so this is a much more complex organism from bacteria, obviously. She would have had her DNA spliced with bioluminescent jellyfish gene. So it seems under blue light, and I think you'd have to sort of look under the fur. It's not like the fur would have been bioluminescent, but the skin of the rabbit would have glowed in the dark under blue light. So that's the idea. But it never, the French state, the laboratory, once they made the work, they refused to release the rabbit. The rabbit was supposed to go back to Eduardo Cac in his home to live with his family in Chicago, but that never happened. And so this work that was originally this idea to have a transgenic rabbit, which was supposed to be the work itself, then became this whole other work about her being freed and released. So he did all these lectures, he put all these posters up, he did an whole exhibition called Free Alba. So it became, and he probably liked this in a sense, although I'm sure he would have liked to have Alba. I think it's still a mystery as to where she is at this point. She's obviously she's most definitely not alive anymore. If she was born in 2000, the lifespan of rabbits are probably like, what, 8 years or something like that, maybe a little longer. So never made it back from the laboratory. And so as far as like the work that elicits the most discussion in class, usually we spend a lot of time talking about this bunny, the ethics of it, in what way it might be interesting, a compelling work, in what way is it a completely irresponsible work. So if you read the other text by the philosopher Elisabeth de Fontenay, the French philosopher, it's a scathing critique of Cac. It's a scathing critique of this type of work. And she makes the point that if he cares about, if this work is about animal ethics, if he cares about the well-being of animals, it would have been much easier and cheaper for him just to adopt an animal, rather than make a new one with a special feature of a spliced gene. So if you're not being generous to Eduardo Cac, you might be in line with Elisabeth de Fontenay's really brutal critique of this type of work. But then it's possible that you actually find the work interesting, and in some sense there's nothing wrong with that. I find the work both compelling, but also disturbing all at once. So sometimes, we're humans we can be cognitively dissonant. We can have two mutually opposing ideas in our minds at once. That seems to be a feature of our lives. So I find this compelling and disturbing all at once. So I'm sure you'll end up writing quite a bit about that one. And then the other work, which comes a little later, is just a year later, is The Eighth Day. And this is another one where you have this. Now you have a whole ecosystem of transgenic life, from bacteria up to like amphibians, and again, people are able to use the blue light, see from remotely from the internet what's happening in the work, as live bystanders are seeing into the work. But people from the outside through the internet who are looking in from the top down, also see the people. So it's like the people, the living people who are inside the exhibition space, they become part of this work. And the other part of the work is, I suppose this is like the advanced part of this work, is the Bioart, this robot that's in there, whose brain is actually made of amoeba cells, transgenic amoeba cells. So the idea is that as the amoeba cells are doing their thing, and they're also mutating or being affected by the blue light, they're basically the brain of the robot in there. Their mutations, their activity dictates the movements of the Bioart, along with the activity of people online who are able to use it to tilt the camera and that sort of thing. So that's the basic setup of the 8th day. So I'm sure you're going to end up talking a bit about that. I would say Gfp Bunny is probably the one to talk most about, maybe Genesis too, but you can talk about all three. And so there are a number of artists that are working in this mold of transgenic art. Another one is Marta de Menez. This is Nature, from around the same time. So Bioart really kicks off in the late 90s. And there's a heyday of it in the late 90s and early 2000s. There's still a lot of Bioart going on today. At SVA, there's a whole Bioart program run by Susan Anker. So there are still a number of artists working in this mold, which is a mix of laboratory, scientific, artistic endeavor. So Marta de Menez in Nature went into butterflies and altered the gene for the expression of their colorations and the patterns. So you probably know that a lot of butterflies. Scientists have been fascinated with the designs of the butterflies because they can be really intricate. They could look like other animals. They either blend into the environment or they look threatening in some ways, right? And there are all sorts of explanations as to why butterflies would have developed these incredible patterns as an evolutionary trait, right? Which would help their preservation, which then they would pass on to their progeny. Right, so that the positive traits would keep getting repeated over the course of the evolutionary life of this line of butterflies. So that's that's a basic premise understanding of evolution since Darwin. And so what this artist does, she went in and altered the genetic code of the butterfly so that the pattern would be altered. And it would only be on one side of the wing. So notice, like a normal evolutionary, if this butterfly would not have been, let's say, tinkered with or meddled with at the genetic level, both wings would have looked like this. But the tinkering creates these other patterns, again, only on one side. And do I have another? No, I don't. There are other examples. So if you're interested in her work, you could just Google her in Nature. And then see these other butterflies that she quote unquote created or modified. So the interesting thing, and I think this is for her, the interesting thing is I think this is why she has these titled as Nature ? Is that are we still within the processes of nature here? Is there a way in which she's biohacked evolution, or is this still a part of the history of the evolution of these creatures? The other thing I know about this work is that the way she did it, the progeny of the butterflies that are altered will not get that trait. So for her, it's important that she hasn't altered like the evolutionary line of these butterflies beyond just this one. And once this one dies, that's it. Like there will never be another butterfly like it, right? So this seems to be an important feature for her work. So this is very much within the realm of Eduardo Kac. So going in and making transgenic creatures. Both Marta de Menez and Eduardo Kac, I think, even though this work is like laboratory scientific work, and I think you could tell when you look at the exhibition of the Eighth Day or Genesis, it almost looks more like a science display than it does a work of art to a layperson, right? And the same, I think, is true of Martha de Benenes. There's almost more of a sciencey but aestheticized version of science in these types of work. But that doesn't mean all bioartists or transgenic artists are working in that way. So I'm sorry if you're having lunch. This is very, very gross. This hunk of flesh sitting on a plate. It's human skin left over from plastic surgery. So skin and then fat cells and so on and so forth underneath. And so this artist, I don't know if I don't know her that well. So I'm probably saying her name wrong, but Jalia Asadi, Jalia Asadi. Email me if you think you have a better pronunciation of this name. I'm not totally sure. But she made an interesting work that's also transgenic. It's called 2.6 grams, 329 millimeters per second, also known as bulletproof skin. So for her work, and this is less aesthetic and slightly more within the realm of utility and even activism, you'll see. So she took human skin that she got from, I'm sure she had some sort of agreement with the plastic surgeon. This stuff probably just gets thrown out. Maybe it's liposuction or something. And I'm sorry if you have a fear of spiders. I should have warned you. Here's a spider. So she'll take the human skin and then make a transgenic human skin. She'll splice it with spider silk. So the webs that spiders use, spider silk, in order to make a transgenic hunk of flesh that has the genetic features, the components of spider silk and of human skin tissue. And the reason you'd want to do this, the reason she does it, is that scientists are still, I think there's been some recent developments, but they're still unclear as to how spiders make this, because it turns out that spider silk is one of, if not the strongest material on Earth, which is pretty wild. It's stronger than steel, like 5 times stronger than steel. But the other thing that's incredible about it is that even though it's stronger than steel, it's actually pliable. It's plastic. So scientists still don't understand how you can have something that's so strong, but also bendable and movable and plastic. So these little spiders have been studied for quite some time. There are all sorts of, you can imagine military uses for this kind of thing. And so what she did is she made this transgenic human flesh. And so, basically, we're talking about a chimera. We're talking about a hybrid between a human and a non-human animal here. Human skin with with spider silk in its DNA. And the idea is that the skin then becomes bulletproof because it's so strong. So there's been some testing. She does testing for this. So this is just a still. Unfortunately, I hate doing this because I always like my PowerPoints to be seamless. For whatever reason, her YouTube video for this is protected, so I can't pull it in any way. So what I have to do is just go to it on my other screen, which isn't too bad on this computer. So it's just a minute long, but it shows you in slow motion a bullet being shot at the skin, and the skin basically taking the bullet, and the bullet comes back out, rebounds backwards, and the skin is intact, right? So let me just show it to you real quick. It didn't stop. I was reading about this. It doesn't stop every bullet. It doesn't stop it just at a certain threshold. So it's not like this would really be useful. I mean, Oh, here's another version of it. I think this is the one where the bullet comes back out. So because the skin is transgenic, with spider silk, it is able to withstand the bullet. Oh, right, this is not the PowerPoint. Sorry, everybody. Let me close out of my Safari and go back to the PowerPoint. Okay. So, I mean, there's a clear sort of activist dimension to this. This is 2011, 2013. So, you know, we're already within this, especially in this U.S., this gun epidemic of mass shootings, and not just in the U.S. 2011, 2013. This is around the time of still one of the worst mass shootings in Norway. That guy Breivik, who goes and kills all these children on an island. So in some ways, this is more of an activist work. I mean, I can't imagine, but activist in still like this more poetic way. I mean, you can't imagine like actually outfitting somebody with the skin and the skin being useful, or somehow, you know, starting to genetically modify children so that their skin will be bulletproof. This is so dystopian and kind of horrifying. These spider silk children walking around. It's just kind of horrifying. So the work, don't see it as, in some ways, I don't think she thinks of it as useful in any way. So it's more of this aesthetic, activist kind of work, which is certainly transgenic, right? Which is certainly an instance of bioart. Here's another work which is not. I didn't know where else to put this because it's an interesting work and also pretty controversial. So it's not transgenic, but it definitely is bioart in the sense that you're now. It could have been in the posthumanist section from last week. You are marrying the human body with a non-human body in a form of cooperation, but because it's influenced by transgenic art or transgenic processes in biology, and because there is kind of a scientific, medical theme to this work, it actually is designed. Revital Cohen is a designer. It is designed for practical medical use. This work, life support. So life support is not being cheeky. It actually really is life support. And so the idea is this designer made, using a greyhound, made basically like an aqualung, where the oxygen generated by the greyhound on the treadmill will go into the lungs of a human in need of air. Right, and so it's a little complex. So I pulled the video of Revital Cohen herself talking about this work, explaining it, and this might be an interesting work to write about, to mention in your responses. So let me just show you this real quick.
Trying to find a more humane solution for this medical technology. Why, if you lose your eyesight, you get this lovely guide dog, but then if your kidney has failed, you get this horrible computer which controls your emotion and keeps you captive. And I found this research about scientists that are making sheep and cows to grow with human blood, and the idea was that the scientists were going to kill the sheep, take the blood, and use it for transfusions. Maybe instead of killing the sheep, you just use it as a life support machine. There isn't a computer which can really imitate a real organ. So this machine has a transgenic lamp with human blood in it, and it helps the person clean their blood. And I was also really inspired by the connection of the dialysis machine as an object which is placed near the bed, and sheep as being symbolic animals to sleep. We count them to fall asleep. Children have them near their beds. So the way it would work is a patient will give their blood sample to a laboratory. The scientists then will make them a transgenic lamb with their own blood, which they will keep as a pet during the day and as a dialysis machine during the night. And they will sleep with this sheep next to their bed, with their blood going out through their veins, through the pumps, into the sheep, clean through the sheep's kidney, and come back into their body clean. And the sheep will urinate the toxins in the morning. There was another machine I designed, which was a mechanical ventilation machine. Greyhounds are already in a way manufactured now for the racing industry. They're being bred by the thousand, sent to racing, and after 2 to 5 years, euthanized when they become redundant. So in my scenario, instead of being killed, the greyhound will be adopted by a doctor who will retrain them to become this ventilation assistance animal. In this case, it's a non-invasive harness that the dog will be wearing. Whenever he's breathing, his lung movement pushes the small bellow, which then pushes the air into the big bellow and into the patient's lung. Just one last word about the treadmill. If you design a machine made out of an animal, where's the on-off switch? Where's the dial? So the treadmill basically works on the greyhound's previous education. When the person needs more air, he presses a button, the rabbit starts bouncing, greyhound runs, person gets more air. A dog obviously can create such a more symbiotic relationship with the patient than any big computer. The greyhound was perfect both on personality and physicality for the job. This chart is just to show that none of these are just kind of crazy ideas that come out of nowhere, but actually a result of a very serious design process with everything being calculated, engineered, checked with scientists and medical consultants. Another thing that my project was meant to do is to make people think of the use of animals as products. I mean, this seemed quite outrageous, but then people eat lamb every day, and greyhounds are products of entertainment, just bred and killed for people's pleasure. And I think as designers, we're reaching a stage where advancements in biotechnology allow us to start thinking of design as something that will break the boundaries between natural kingdoms. Material is not going to be only metal, wood, plastic. How about tissue, animals, people? The goal of my work is to make people think of new technologies, on how they might penetrate their lives, how will they change the way we live, the way we behave, the way we think, the way we perceive ourselves.
So her last point is important. One of the reasons why this is not technically a transgenic work of art, that she's influenced by transgenic biotechnology. There is something of a collapse between the organic and the non-organic, or the animal and the machine here, the medical apparatus, which seems to be a result of this type of bioart, where we start to, if last week we talked about the collapse of nature and culture. We live in more of a post-natural world where the division is very blurry. Here in bioart, we're talking about a collapse between what's organic and what's inorganic, what's animal and what's machine. That they can be melded, they can be fused, and if we get back to the idea of actually tinkering with genetic material, life itself, the substrate of life itself, becomes almost like a machine that you can sort of tinker with and play with and reconfigure. Right. So it's in some ways the 20th century is we have a whole radical reconceptualization of what life is, animal and human life and plant. Right. So that major discussion, the reorientation, reconceptualization of life, is something that's very much a part of this work of bioart, of transgenic art. And so while this is not technically transgenic, it does sort of betray that collapse between animal and machine. So weirdly enough, getting back to sort of the Cartesian conception of the animal that we talked about a few weeks back, of the animal machine, but in this case towards therapeutic ends. And I'd be interested to hear what you think about the ethics of this work, if you choose to write a little bit about this work in your response. And then the last part of what she says in her talk is interesting because it sends us to our final, the last part of what we'll talk about for today, and that's the idea of sort of going in and working with tissue and creating new tissues through these technologies. And so there's a painter, Alexis Rockman. We'll see him again when we talk about the aesthetics of extinction, and maybe the larger our session on ecology and environmentalism and climate change, because he's done a number of large-scale paintings that bear on these themes. This is an early work of his called The Farm, and he says that we should actually read it from left to right. And so if you do read it from left to right, you go from, let's say, more traditional forms of life. So here you would have a rooster that looks like a rooster on the post of this fence. You have a pig, you have a steer in the back there. It's as if we're moving like in the 20th century here, a whole history, because here you have the double helix. So maybe this is, when was it, in the 50s or 60s, where you have the discovery of DNA. And as you move forward, it's like you're moving forward in time. So you have a little mouse down here. You're moving forward in time, and you're starting to see these more and more developed ways of actually altering animals. And in this case, often towards, let's say, human ends, or human ends of production, of economic production, capitalist production. So you go from, again, a larger pig, a larger cow. In the 20th century, animals get bigger and bigger and bigger, to this cow that looks like a big rectangle, to these tomatoes, and these are all not the cow, although cows have been genetically modified in a number of ways, including with the ability to bear and produce more milk, and often in pretty dire consequences for the well-being of the cow herself. And then produce being shaped in certain ways. There's this famous watermelon that's been bred, not transgenically, but it's been bred to be like a box, so that it can be stored more easily and more efficiently than the regular, oblong, rugby-shaped watermelon. So you have here with these tomatoes that just fit neatly within the basket. You keep moving forward. You have this mouse with an ear. This is an actual mouse. So this is also the history of scientific breakthroughs in the 20th century. This is the Visconti Mouse from 1996. This is not a transgenic work. He didn't go in and it's a mouse with an ear on its back. It's really, I don't know, I find it very disturbing. He didn't go in and change. The Visconti, there were these two brothers, these two scientists, and that's why the mouse is called the Visconti Mouse. It's not a transgenic work, so they didn't go into the embryo and change the DNA so that the mouse would somehow grow an ear, like the Oncomouse. The Oncomouse would actually develop cancer through intervention in the embryonic stage and transgenic manipulation. This was basically more fused together. This ear was grown, this tissue cartilage ear was grown, and then attached, and it cultured it attached itself to the mouse. So you remember last week we looked at Stellark, that artist who put an ear on his arm. Same process, right? So the tissue would blend into the tissue. So this is melding two tissues together. And the Visconti mouse, like Dolly, like the Oncomouse in the late 80s and in the 90s, was very much wrapped up in these debates, the bioethical debates of doing these sort of experiments. And for me, unlike Oncomouse, the Visconti mouse is completely unnecessary. So I think we have to make a distinction between the two. It's unclear. It's possible, and we'd have to read more about it, but it's unclear how the Visconti mouse actually furthers the human well-being. So that's the Visconti Mouse. And so you'll see it. Rockman has actually placed it in the painting in midway point here. You have parakeets. You have, again, further development of poultry all the way up to when you get to here, you have the Federalist chicken that I talked to you about before. And you'll notice this chicken also has, instead of 2 wings, you have a much more profitable body if you have 3 wings on each side, so that you have 6 chicken wings, basically, right? I don't think this has actually been done, though it's certainly true that chickens have been bred and modified to have larger breasts, so that they would be sold more. The more weight, the more you can sell it for. So that's certainly real. Then you have like this pedigreed dog over here, and then this massive pig. Pigs have been bred, and in certain cases genetically modified to be bigger. And then also, and this is recent, pigs have been genetically modified to grow human stem cells to grow human organs. So this is why Rockman has, this is a kidney, these are lungs, and this is a heart inside the pig. So the pig will then be, as it grows, as it develops, it basically just becomes an incubator for organs that then can be taken out. It can be killed and taken out to be used for humans that might need these organs because of whatever diseases they might have. So Rockman's painting, what a complex history in the 20th century of the ways in which non-human animal life can be manipulated, in some cases genetically modified, in order to be used both in scientific experimentation, in the medical field, but also simply in the economy, in this large-scale agricultural economy that privileges more and more production and more efficiency and more profitability. So this is very much a part of the history of bioart that you don't really see. Like if you were talking about Eduardo Kac, if we go back to really any of the works we've looked at so far, this really doesn't seem to be part of the discussion. But there are artists, and we'll end with these. There are artists that have worked on this question that are in bioart. So Oren Katz and Yonat Zur, this is in the mid-90s, 96 I think is when the Tissue and Culture Art Project first gets underway, founded by Oren Katz and Yonat Zur in Australia. They're Australian artists. And they started making work on what they called the semi-living. So they would produce tissue, often transgenically, that would then grow on its own, or it wouldn't grow on its own. It would have to be nurtured by these apparatus that are developed by the artists. And why they call it semi-living tissue is that it's somewhere between living and dead. It's somewhere between like an amoeba or bacteria and a fully fledged creature like a human or a rabbit. It's somewhere in between. It's a piece of tissue that does grow. So this is a work called Victimless Leather, and a lot of their work actually falls within the rubric of developing tissue that would normally come from an animal that had to be born, live, and die, or be killed. Their work would actually develop the tissue. It would be real. This is real tissue. This is real flesh being that does actually grow and grow and grow. But it wouldn't necessitate like a sentient animal. It's only the tissue itself. So this is why it's semi-living. It's living in the sense that it is going through organic growth, and not organic growth in the sense of a plant, but organic growth in the sense of animal cells, right? Animal flesh. But it's not a sentient. It's not a sentient entity, right? And so this is Victimless Leather from 2004, which MoMA had. MoMA was showing, and it grew so fast. You can tell this is the aesthetics of the laboratory, right? This looks much more like a science experiment. It's this very small little leather jacket that they made out of transgenic flesh. But the jacket grew so fast, it started to break out and interfere with its own. It has to be fed nutrients. It has to be they have to be daily cared for, and they can't. It has to be completely sterile. These are very fragile tissues, these semi-living tissues. So if it comes into contact with a non-sterile environment, certainly if like someone would touch it, the bacteria on our hands, on the surface of our skins are much more powerful than the tissue cells in the work itself, and so it would just destroy. It kills the semi-living tissue. So MoMA had to actually take it down early. So that's victimless leather, and I think you can see why they called it victimless leather. Genuine leather, not imitation leather, does come from skin of various sources, mainly cow. Then the year later the year before, they did a work called Disembodied Cuisine, which I think is kind of fascinating. Also part of the Tissue and Culture Art project. So what they did, they set up a laboratory. They have to, because these things don't really travel well. These tissue cultures, they're so fragile, and they have to be kept sterile, and they have to be fed and cared for daily. And that's another part of their work. This idea that this fragile living thing needs to be taken care of. So what they did is that inside of this laboratory, they took cells from a frog, or from more than one frog, and developed. This is in France, and developed what they called semi-living steaks. So basically, what they did, they took the cell from the frog, they cultured it, they cultured this tissue, so that it would grow to the size of basically like a steak that could be used in cooking. They also grew herbs, and then ended up cooking the frog steak. And you'll notice here there's a dining table. So the work also included people that were invited. They can come and sit down and eat the frog steaks as part of the work. And you'll notice, so it's right here. There's like a terrarium that's looking out over the dining room, which is completely sterile, which is completely enclosed. And the frogs themselves, who are doing well and fine, they're in the terrarium, and they look on as people are eating the steaks that were made from one of the cells of their, like a biopsy from their bodies, right? So this is why it's called disembodied. I have a typo there. This is why it's called disembodied cuisine. And I love the fact that there's this bronze Venus de Milo in the terrarium, because again, you have this whole history of art and bodies sort of collapsed here in this work. And so the Tissue and Culture and Art project, they've done a number of different works like this, but you can tell they're working both within art, within bioart, but they're also interested in much more upfront, much more clearly and less ambiguously than someone like Eduardo Kac. They're much more upfront about its uses in ethical debates and debates of food, right? Of food production, which historically has involved animals. So, is there a way to rethink food production that comes from animal sources, so that you don't actually harm an animal for the final product? That seems to be it's definitely what this work is about, and the Tissue and Cultural Art project is about. And this is 2003. It's quite early on, but you may know, or you may have heard, in the past 5-10 years, there's this idea of cultured meat, or it's also called clean meat or cell-based meat, that this has now become a reality. So they were early within this. This is an interesting moment an interesting example where artists, in this case bioartists, are before the curve in these sort of developments. So what they were doing in this work in 2003 has now actually become a reality. So you have a number of companies. One of the big ones, you should probably start seeing them in stores, is called Just Meat. So basically, you take a biopsy from an animal, and the animal can then just, you could have these animals in sanctuaries, and they live their lives. They die a natural death. You take the stem cells from the biopsy. The stem cells can then be grown, and they can become muscle tissue, and then this can then become material for conventional food, right? So this is an example of beef patties that have not come from that do not come from a dead body. They come from a living body that then generates what Oren Katz and Yonat Zur would call semi-living tissue, right? Semi-living tissue that can then become the conventional material for the food industry. So this would actually be a huge win from an ecological perspective, because in our last class we'll talk about how detrimental animal agriculture has been for the environment and what a major player it is in global warming. It's also an advance within animal ethics, because now all of a sudden, if flesh that's consumed by humans doesn't come from living animals that have to live and die in usually pretty horrible circumstances, then all of a sudden you have a win for ethics, for ethics. The issue, of course, is that we're coming to find that materials like this, most animal products, things from milk to especially red meat and processed meat, these are in fact really not good for human well-being. So things like heart disease, things like certain types of cancers, diabetes, type 2 diabetes, autoimmune disorders. Like, we are coming to find, like we did with tobacco, like we did with cigarettes in the middle 20th century, that actually these are not, these are actually causing serious harm to people, because if this is their daily fare, if people are simply eating animal-based products 3 times a day, 7 days a week, through their whole lives, that actually it's actually pretty clear that it's causing human harm. So while cultured meat, while cell-based meat and the semi-living tissue would definitely be a win from an ecological perspective, from environmentalism, which is why it's growing. So the markets for plant-based meats and now these type of clean meats are really growing, because everybody is more or less on board that climate change is a serious problem. So it's part of that solution. It's part of the solutions of animal ethics, and the majority of people, once they understand what animal agriculture is all about, they become uneasy about it, right? They think, oh, well, this might not be ethically justifiable. These are all wins for that. From a nutritional standpoint, it still might not be the best thing. So maybe in the future, these kind of things will be luxuries that people have, but certainly not something that they would have every single day. But that's for a whole, debate and discussion about human nutrition and how it's evolved, and what seems like what's good for humans and what's not good for humans. That's a whole huge discussion that we'd have to have, which I'm not really qualified to give. But there's all sorts of information out there, researchers and scientists that are working on these things, and it's becoming part of the popular discourse. So that police siren in the back is telling me that I'm in my Harlem apartment, that you're probably hearing some outside noises, but I think you hear me pretty well. It also tells me that let's call it a class. We went through quite a bit there, which is good. I'm just going to leave you with some questions as you're doing your reading responses to the CAC reading and to the video, and maybe interspersing some of the works that we looked at today. I just want to leave you with some questions, the ones that I think are important to think through. Obviously, you don't have to write about all of these, but write about one or two, whichever one you think is most interesting. And some of this comes from that Elizabeth de Fontenay reading, the second one I recommended but didn't require you to read, where she just blasts Bioart. So most generally, like, what do you think the ethical implications are of such art practices? When do ethics come in what ways? That's an interesting question. Then the other interesting question is, do artists like Kak, are they really creating something new? Are these transgenic techniques really creating something new? Or is this more like bricolage? Are they really only kind of cobbling together pre-existing materials? And is it hubris for Kak to think that he's actually created something new? And you'll note he has a lot of his work has these theological undertones of creation or superseding creation, usually Christian, Judeo-Christian creation stories. The other interesting question is, do you think it's possible for an artist to be as well versed in art history, in art practices, and in biology? Or is it inevitable that the artist will either be really good as a scientist, really know the science very well, but then be completely unaware of or not trained enough to be able to really make a contribution to the history of art through these transgenic works? Or vice versa? Do you think that someone like Eduardo Kak is well versed in art history or in contemporary art, and is making contributions to contemporary art, but from the perspective of a biologist, the stuff that he's doing is actually inane, child's play? That these are actually just simple techniques at this point, and it's almost like patronizing for a scientist to contemplate it, right? So this is one of the questions that Elizabeth de Fontenay talks about in her reading, and I think you can tell which side she falls on. The other and final question that we might ask is, do you think that bioart can be a critique of instrumental biotech? And by instrumental, I mean like using these biogenetic technologies just to make a profit, just for human-centered research or commerce. Do you think it's possible that this work can be a critique of that? Or do you think it's just it serves as an unwitting advertising for biotech? It's just kind of a glorification of these newer biogenetic technologies. So those are 4 questions that are pretty pertinent. You might want to think through one or two of them in your response. But of course, I'm kind of more interested in just your own personal critical take on these works and on the reading. So I look forward to getting your reading responses. And again, you can have those in, if today is a free day for you have plenty of time, go ahead and get it in today. If not, you can turn it in tomorrow, on Thursday, or even Friday is fine. From these remote, now that we're doing all this remotely, I won't be a stickler about getting things on time on the day of the class. It's totally cool. You can take 1 or 2 days to work on it, and then really think through and provide a really nice response. So until next time, stay well out there.